Now, question number 26. Which of the following cell organelle control intracellular digestion? Digestion means conversion of large molecule to the smaller one, and that too is with the help of hydrolytic enzyme. What is hydrolytic enzyme? These enzymes are actually responsible for the breakdown of complex molecule. For example, C12H22O11 is sucrose. If we add sucrase enzyme and water molecule is required, this water molecule is responsible for the lysis of the sucrose into hexose sugar, that is C6H12O6 plus C6H12O6. See, this activity is hydrolysis. Why? Lysis of larger molecule into a smaller one by the addition of water. Lysis, that is the splitting. Then hydro, that is pertaining to water. Water molecule is added, then the complex molecule becomes into simpler form. Such enzymes are known as hydrolytic enzyme. And these enzymes in cell, they are found in lysosome. They are found in lysosome. So, lysosome is a cell organelle which is covered by one membrane reported by the duve. It contains hydrolytic enzymes, and these enzymes are acid hydrolases. Enzymes of lysosomes are acid hydrolases. That is, they are functional at acidic pH. They are effective in acidic pH. So, they are acid hydrolase. So, final answer that is concerned with the digestion, that is lysosome. The lysosome is a cell organelle which is single membrane bound cell organelle rich in hydrolytic enzyme. These enzymes are called acid hydrolases because they are functional at acidic pH. Plastids, they are found in plant cell. They are pigment bearing cell organelle, and one of the very good or most important plastid, that is chloroplast, green color plastid. It performs the process of photosynthesis. Peroxisome, a microbody. Peroxisome is microbody commonly found in C3 plant. This microbody is commonly found in C3 plant and related with the photorespiration. Peroxisome, single membrane bond microbody. Second, concerned with the photorespiration process. It is associated with the photorespiration, that is glycolate metabolism process. And glyoxysome, these are also microbodies. They are especially found in oil seeds. Convert non-carbohydrate into carbohydrate. Non-carbohydrates into carbohydrate, that is gluconeogenesis. But for this question, the answer is lysosome. The answer of this question is lysosome. Then, which one of the following is not the hydrolytic enzyme? Lipase is hydrolyzing enzyme. Hydrolyzes lipid-like substances or fatty substances. Sulfatase, sulfate esters. They are effective on sulfate ester. Phosphatase on phosphate bond. But aldolase enzyme is not a hydrolyzing enzyme. It is desmolizing enzyme. Aldolase is desmolizing enzyme found in mitochondria, etc. Desmolizing enzyme. Aldolase is desmolizing enzyme responsible for the condensation as well as cleavage. Responsible for the condensation as well as cleavage. In a respiratory activity, one of the compound known as fructose 1,6-diphosphate. Fructose 1,6-diphosphate. With the help of aldolase enzyme, it breaks into 3-carbon compound 3-phosphoglyceraldehyde plus dihydroxyacetone phosphate, DHAP, that is acetone phosphate. So 6-carbon compound undergoes cleavage with the help of enzyme aldolase. This enzyme is also responsible for their combining. That is, if required, then this enzyme works in reverse direction and combines 3-PGL and DHAP into fructose 1,6-diphosphate. So this is not the hydrolyzing enzyme. Aldolase enzyme is desmolizing enzyme. pH for the lysosome, that is acidic. The enzymes of lysosomes are effective at acidic pH, and that pH is roughly 4.5 to 5. That is, in this range these enzymes are functional. So these are called acid hydrolases. Enzymes of lysosomes are known as acid hydrolases. They are called acid hydrolases. One more question which was asked in the previous medical entrance exam: if lysosome ruptures within a cell, the answer, cell will die. The answer, that is, cell will die. So lysosomes, if ruptures within the cell, cell will die. Then one more question associated with the lysosome. Lysosome is known as suicidal bags. Of cell. Why? They are also concerned with the autophagy. Lysosomes, that is autolysosomes, they are concerned with the autophagy. Suppose this structure is non-functional in cell. It may be anyone's, for example, mitochondria. Primary lysosome surrounds this structure, and they combine and finally digest this structure. These lysosomes combine one and with the other one, and finally their enzyme digests this useless structure of a cell. This is autophagy. So lysosomes, they are also termed as suicidal bags of cell. One of the activity, that is autophagy, is seen. Under pathogenic condition, they may digest healthy structure also. Lysosomes, they exist in 4 forms. Number 1, primary lysosome. That is lysosome with digestive enzyme only. That is, enzymes are in inactive stage or precursor stage in primary lysosome. This is first one. Then secondary lysosome, they are called heterophagosome. Secondary lysosomes are also called heterophagosome. That is when primary lysosome combines with food vacuole. Suppose this is food vacuole and this is primary lysosome. So when primary lysosome combines with the food vacuole, that structure is known as secondary lysosome. Then third type of lysosome, lysosomes are involved in the digestion After digestion, digestive remains are present within the lysosome. Those lysosome which contains digestive waste, they are called, this is digestive waste. Such lysosomes are known as residual bodies. These are called residual bodies or tertiary lysosome. And last one I have already described, that is autophagosome or autolysosome. They are responsible for the autophagy. Main role of lysosome is intra as well as extracellular digestion. Intracellular as well as extracellular digestion. The 29th question is again concerned with the lysosome. That is suicidal bags of cell. In general, that is lysosome. Lysosome are called suicidal bags of cell. Vacuolar membrane, large central vacuole is found in plant cell. This is a plant cell and it is outermost covering, that is cell wall. Next to cell wall, membrane is present. This is membrane, cell membrane. It is next to cell wall. This is next to cell wall. Cell membrane is present. Then in mature plant cell, large central vacuole is present. This is the large central vacuole. It occupies approximate 90% space of cell. And the bluish color structure which surrounds the vacuole, that is vacuolar membrane, and its name is tonoplast. Vacuolar membrane is called tonoplast, and the content of the vacuole is known as cell sap or tonoplasm. This is known as tonoplasm or cell sap. Then plasma lemma is common membrane just below the cell wall. It is also called plasma membrane. Karyotheca, karyon, that is nucleus. The third one, that is for nuclear membrane. Karyotheca is actually nuclear membrane. Covering of nucleus is known as nuclear membrane. And one speciality with the nuclear membrane, that is it is double and porous. Nuclear membrane is double and porous. So there are two membranes. They run parallel but they join together only at nuclear pore. This area is nuclear pore. So nuclear membrane or karyotheca is porous. Nuclear membrane or karyotheca is porous. And second, double, question medical. It is also question medical. Then tonoplast I have already described. For the 30th question, the answer is 4. That is vacuolar membrane is known as tonoplast. In plant cell, the vacuolar membrane, I have already given that clue, that is 90% space is covered by. Then now 32, size of mitochondria. This is the size of mitochondria. Some cell organelles are there whose size is generally asked. For example, plastids, chloroplast. Size of chloroplast is also important. Size of lysosome importance. Size of lysosome, size of chloroplast. So here the mitochondria, its size is 0.2 to 1 micrometer in diameter, whereas its length is 1 to 4.1 micron. Chloroplast is comparatively larger, 5 to 10 micrometer in length. Next structure. Cells which require large amount of energy would like to contain relatively high number. Best answer, mitochondria. Mitochondria is actually concerned with the respiration. This structure is concerned with respiration. Mitochondria is concerned with the respiration. Second, in mitochondria, electron transport system is present. ETS is present in mitochondria. And when electron transport system is functional, mitochondria generates number of ATP. Mitochondria generates number of ATP or energy units. So these ATPs are synthesized as well as available from mitochondria. Mitochondria is a structure which is responsible for the formation of energy units ATP, as well as it also supplies these energy units for other vital activities. So mitochondria is known as powerhouse of cell. It is called powerhouse of cell. Mitochondria is known as powerhouse of cell. Why? It generates energy units ATP as well as supplies them for the other vital activities. So if mitochondrial number is larger, then the amount of energy is also available comparatively more. So here the answer is mitochondria. Golgi body concerned with synthesis and secretion. Chloroplast is concerned with the synthesis of food material by photosynthesis process. Centrioles, they are non-membranous structure, mostly in the animal cell. They are related with the formation of a spindle. Centrioles are concerned with the spindle formation. Centrioles commonly found in animal cell, first one. Second, they are non-membranous structure. Third, they are responsible for the formation of a spindle. They are responsible for the formation of spindle during cell division, and that spindle responsible for the poleward movement of chromosome. The role of chloroplast, photosynthesis. What is the role of chloroplast? Photosynthesis. Golgi body role, that is synthesis and secretion. Synthesis and secretion. Marker enzyme of mitochondria. Question pre-medical. This is again question of pre-medical. Marker enzyme of mitochondria. Answer first is correct. That is succinate dehydrogenase. This enzyme is located in electron transport system or electron transport system, and it is a part of complex number 2. Complex number 2. And the oxidizer associated with this enzyme is FAD, flavin adenine dinucleotide. Oxidizer associated with this enzyme is FAD, that is flavin adenine dinucleotide. Flavin, A for adenine, dinucleotide, dinucleotide. This is oxidizer associated with this enzyme. So marker enzyme of mitochondria is succinate dehydrogenase. And mitochondria specifically stain with the help of genus green B. These are the few questions which were already asked in the examination. Genus green B. Genus green B is vital dye for mitochondria. Genus green B is vital dye. Vital dye, that is specific stains which stains that particular structure without affecting its physiology. Those dye which stain a particular structure without affecting its physiology. Such dye are vital dye, and for mitochondria that is genus green B. For mitochondria that is genus green B. Then one more thing concerned with the mitochondria, that is mitochondria is double membranous structure. This is outer membrane, and second speciality with the inner membrane. This is a question for pre-medical. That is, inner membrane has several folds. These are called cristae. These folds are known as cristae. Folds of inner membrane, these are called cristae. And on cristae you see oxysomes are present. Oxysomes are present. This is the structure of mitochondria in brief. Structure of mitochondria in brief. That is, these are the oxysomes or F0, F1 particle. These are oxysomes or F0, F1 particle. Now, proteins are stored in plant cells. Plastids are present. Plastids are found in plant cell. Eukaryotic plant cell, not found in prokaryote, even in animal cell. Plastids, pigment bearing cell organelle. They are pigment bearing cell organelle. Pigment bearing cell organelle. There are three types of plastids, basically with respect to the pigmentation. Number one, leucoplast. These are colorless plastids. Leucoplast are colorless plastids. And leucoplast mainly concerned with storage. First one, amyloplast. First one, amyloplast. They store amylum or starch. That is, starch storing leucoplast are known. Those leucoplast which store starch, they are amyloplast. Starch is stored in amyloplast, and amyloplast are type of leucoplast. Second is aleuroplast. That is, proteins are stored. In this structure, proteins are stored. Third is elaioplast, in which fats and oils are stored. Elaioplast store fats and oil. So, elaioplast fats and oil, aleuroplast protein, but the proteoplast is also given. So, better one is aleuroplast. Better one is aleuroplast. Then these are the different types of leucoplast. Then second type of plastids are actually chloroplast. Second type of plastids are chloroplast. Green colored plastids. Green colored plastids are called chloroplast. Green colored plastids are termed as chloroplast. Their shape varies in algae, green algae. For example, in Chlamydomonas, this is a Chlamydomonas, the shape of chloroplast is cup shaped. This is the shape of chloroplast, it is cup like. And within this chloroplast, one more speciality is there, that is presence of pyrenoid. So, one question is also related with this one, that is pyrenoid. Pyrenoid is found in chloroplast of green algae. Pyrenoids are found in chloroplast of green algae. proteinous granular structure with which is covered by starchy sheath. Proteinous granular structure covered by starchy sheath. This is the starchy sheath. The role of pyrenoid, they are associated with the accumulation of starch. Then in higher plants, they are biconvex or planoconvex. Biconvex or planoconvex, covered by two membranes, in which inner membrane is less permeable in both the cases, that is mitochondria as well as chloroplast. Outer membrane is comparatively permeable and having porine proteins also. And this chloroplast perform photosynthesis. And chloroplast, one more speciality, they have a disc-like structure known as thylakoid, and these thylakoids are arranged one and above. And they constitute granum. So one question is possible, that is membranous disc-like structure found in chloroplast. These are known as thylakoid. They have photosynthetic pigment. Photosynthetic pigment is distributed over the membrane. So photosynthetic pigment, location of photosynthetic pigment on chloroplast, that is thylakoid membrane. And chloroplast converts light energy into chemical potential energy by the process of photosynthesis. And the last type of plastids are chromoplasts. They are colored plastids. They are colored plastid but other than green, and they are found in colorful regions of plant like flower petals. And finally, leucoplast, chloroplast, and chromoplast. These are convertible structure. This is all about brief idea about the plastids of plants. And for this question, again, best answer is aleuroplast. They store proteins. Aleuroplast stores proteins.